Welcome to Future Talk. On today's program, we're going to talk about one of the most rapidly developing areas of science and one with enormous potential to change our future, and that's the science of genetics. I have two guests. Michael Snyder, PhD, is chairman of the Genetics Department and director of the Center of Genomics and Personalized Medicine at Stanford University. He's a leader in the field of functional genomics and proteomics, which is the science of proteins. He's created a number of new genome mapping technologies and is co-founder of several biotechnology companies, including Protometrics, Affomix, and Personalix. Russ Altman, MD, PhD, is professor of bioengineering, genetics, and medicine, and former chairman of the Stanford Bioengineering Department. His primary research is in the application of computing technology to determine how our genetic traits influence the way our bodies respond to medicinal drugs, which could open the way to much more personalized medical treatment. He's also chairman of the Food and Drug Administration Science Board, which advises the FDA Commissioner. Gentlemen, welcome to the program.
Thanks very much for having us here. Thanks.
Mike, let me start with you. Just to begin at the basics, could you say a few words about what the human genome actually is and what's in it?
Sure. Your genome is your collection of DNA, which is 6 billion bases, 6 billion building blocks, if you will. And each of your cells has this DNA. It really is your instruction manual for how you go from a single cell when you were first started out as a tiny fertilized egg into a human being with 100 trillion cells. Your DNA is what determines that.
Does it just code physical traits like height and eye color, or does it have things like personality and intelligence, intangible things?
Yeah, it does all of that. It does, again, your physical characteristics like your eye color, the kinds of food you digest and things like that. But it's also many of your personality traits are determined by your DNA. They're also determined by environmental influences as well, these various traits.
So what's the state of knowledge right now? Can we say that we can look at a genome and look at every gene on that and know what that gene does?
Well, this is a huge area of active research. We're trying to understand all the details of how your DNA determines you to become like you, and Russ's DNA makes him come out like Russ, and every person becomes the way they are. But perhaps one of the most extraordinary areas that people are working on is trying to predict disease risk from your DNA, what kinds of diseases you might be susceptible to attain in your lifetime, and even things like how long you might live, one hopes to learn from your DNA.
You know, there's a fascinating interplay between your genome and the environment, and we're just starting to unravel that. We know that as you live through your life and are exposed to external factors, that this can modify the way the genetic code gets ultimately interpreted. But we also know that identical twins look identical. And so we know that the DNA has very powerful influences, but that the environment comes in and also does make modifications or some of the choices that need to be made during development.
Now, how do you go about doing genome research? Do you take human cells and take them apart with tiny tweezers under a microscope?
Not exactly. You basically will take, for example, your spit. You'll be able to get your DNA from your spit, and you put it on these incredibly expensive machines. They cost about $800,000 a piece. But you can break it into little bits, and then you actually determine the sequence. And what that means is determining the order of each of the chemical building blocks in DNA. And when you know the sequence, you can see basically how they're ordered and what the difference is from one person to the next. That we have subtle differences. It's only one in about every 1,000 bases or so that are different between you and me. But those differences make you look like you, and me look like me, and Russ look like Russ.
What are the most difficult technical obstacles to solving the genome? What is the hardest problem you have to work through to understand what's going on in the genome?
You know, it used, excuse me, Mike, it used to be that just getting the sequence was a big challenge. The first ones were solved in around the year 2000 at the cost of multiple tens of millions of dollars. Now you can get your genome for $3,000. So actually measuring the DNA is not the problem. The problem is that gives us a parts list, and we need to understand how the cellular machinery reads that and makes decisions to differentiate a cell into a liver cell or into a muscle cell or into a brain cell, and then how those cells know when they should do different functions. And that's the current big challenge.
We have a few slides that might illustrate what we're talking about. Can we see the first slide, please? Okay, so what are we looking at here exactly? Either of you?
Yeah, so at the top is the nucleus where your DNA is housed. So all your DNA is housed in a tiny little package called the nucleus. And the DNA is actually huge. It's, as they say, it has 6 billion of these bases in a row. But actually the part that encodes the genes are the things that make your proteins, that make your liver cell, and your eye cells, and your skin cells. We have 20,000 genes encoded in our DNA. It's about 2% of our DNA. And each makes a different protein that makes the different cell types different from one another and lets them carry out their specialized function.
And the protein is basically the stuff of the cells. That's what the cells are stuffed with, the proteins?
Yes, these are the proteins, for example, in your muscles have contractile power, and that's why you can contract your muscles. Proteins in your eyes can detect photons, and that's how you see. Photons in your brain can transmit electrical signal. Proteins in your brain can transmit electrical signals. So they're an amazingly diverse set of what they call biological macromolecules. Macromolecule because they're huge, made out of thousands of atoms and incredibly complex and intricate.
Now, do these act like little machines where you press it and it responds automatically, or is there some kind of intelligence going on here?
Well, they're machines, definitely. For example, your proteins are what digest your food. The proteins in your gut will digest your foods, and they'll react automatically. But in order to make something complicated like a brain, the proteins really have to be coordinated in their activities so that all the cells in your brain can behave in a certain fashion so that you can think and react. So it's all determined by biochemical activities, but there's a lot of intricate coordination that needs to occur to be properly orchestrated to do the complex things that human beings do.
They are, interestingly, these molecules, they're large, and they are somewhat stochastic. That every one of them doesn't behave exactly the same because of thermal fluctuations in the cell and the exact concentration of the ions in the cell. Interestingly, in the last 10, 20 years, we've been able to make what we call single cell and single molecule measurements. And when we look at these individual molecules, we have an amazing ability to see for one protein, what is it doing, and how long is it doing it, and when does it stop. And these new measurements are giving us a whole new window into the detailed ultrastructure of how the cell works.
Is there some kind of regulatory mechanism? Do these genes turn on automatically? Because the genes are a blueprint, but blueprints don't build themselves, or do they?
Mike's the king of regulation.
Yeah, I mean, they all, again, have to be intricately controlled so they're turned on in the right cells. So you want, for example, your digestive enzymes to be turned on in your gut and not in your brain. You want your nerve cells and the things that control your synapses that let you think. You want those turned on in your brain, those proteins and things. So it's a very complicated machinery. We still don't understand how it works. And it obviously takes not only the hardwiring from our DNA to make this happen, but it also takes the environmental cues that turn things on as well. When you are exposed in an adverse environment, your cells will react accordingly. And a lot of that's controlled at the level of your genes.
We have some other slides. Can we see the next slide, please? Okay, so this is sort of a triangle indicating studies from simpler to more complex.
Yeah, do you want me to do this one, Russ?
Sure.
Okay, so this is following, actually, it's also information flow. So that's your genes at the top. Your genome is your collection of genes. And then function is how they work. And then the next slide is they make proteins. Proteomics is the study of all your proteins. And lastly, metabolomics is the study of all your metabolites, which are produced from your proteins. So each actually comes from the other, and you go from DNA to making metabolites that basically carry out all the little functions in your cell.
So is one of those steps the one that's the main focus of activity now? Do you work on the whole ladder at once, or do you try to pry out the secrets of one? And
it's becoming a little bit specialized, but that measurement of the genome that happens at the top, that's under control now. That's very digital. We can write it down. It's A, C, T, and G. Those are the 4 building blocks. And you can just write 3 billion of those down. They can fit on your iPhone or on your music player. That's not a problem. As you get to those other layers, the protein, the metabolome, the transcriptome, what happens is this very digital DNA signal becomes what you might call analog. It becomes a continuous and modifiable. The genome is pretty much the same for your entire life. The transcriptome, the proteome, the metabolome, those can change from hour to hour, minute to minute. So the real action, I'm a physician, the action in terms of when I see a patient, what's going to be different is their metabolomic profile or their proteomic profile, while their genome remains constant. So it's understanding that dynamic, changing environment that is the key challenge right now. And Mike might want to tell you about some. He's been one of the most measured people in the world because we've begun to actually measure his proteome, his metabolome. I don't know if you want to mention that.
So you test on yourself.
Yeah, we do. Okay. So, I mean, really, where the field is going, we're trying to study our genes, our DNA, to be able to predict human disease. So this is how the project started. We wanted to actually start studying DNA of healthy people and try and predict disease risk. So we actually started on me as the guinea pig because that seemed pretty safe and something I could do without worrying about traumatizing anyone besides myself. So we basically started by actually determining my DNA sequence and then trying to predict the diseases I was at risk for. At the same time, we decided to do a very detailed profile where we would actually look at all the molecules in my blood, my proteins, my metabolites, actually my RNA, which encodes for the protein. So we're actually studying all of my molecules. We make billions of measurements right now on my blood and actually in other parts of my body to be able to see in detail. It's like getting an IMAX movie of your molecular physiology. And we're doing that so that we can see what happens when people's disease might come on. Now, what happened in my case was we sequenced my DNA, and surprisingly, I was predicted to be at risk for diabetes. And as we were going along in the study, about 9 months in, it turns out I got diabetes. My sugar shot through the roof, and we caught that because we were making these billions of measurements in my blood so we could see my glucose go astray. We actually caught it right away, so I in fact changed my lifestyle, meaning I started eating better and I started exercising more, and I gradually brought my glucose under control. But we've been doing all this so we can both understand in detail the physiological changes when someone gets disease, but also the goal is to be able to catch disease early and ideally predict disease and prevent it before it even happens in the first place. That's where we see genomics going in the future.
So it worked in this case, a good case study. We have one more slide in this series. Can we see that next slide, please?
Yeah, in a sense, that's me. So that you can think of that as me in the middle, measuring my DNA in the bottom left part there, and then we're measuring all my proteins and my antibodies, which are things that are my defense system against viruses and bacteria and evil pathogens. RNA, which encodes for proteins in the upper right there, and metabolites are on the left. So we're actually making measurements of all of these things. Again, many billions of measurements on all my molecules in my blood, and actually we do it in my urine and other places as well.
You know, this sounds like science fiction, but this is what we're headed towards in the clinic. Because as a patient comes in, right now we always draw blood tests, but it's very primitive. It's just a couple of things that we know how to measure because we've known how to measure them for 50 years. I think the importance of what Mike was just talking about is this gives us a window into the kind of measurement we're going to be making of patients in the next 20, 30 years. We're going to have a much higher resolution of the dynamic changes that this patient's physiology is undergoing. And the great thing about this story is we're going to catch disease early when it still might be intervention compatible. And that's, again, as a physician, that's the most exciting thing is I don't want a patient to come in where everything has already happened and I'm just in response mode. I'd like to be able to make measurements in real time so that we can proactively alter the course of a disease and maybe even nip it at its bud. So this is very exciting, and I don't think you should think about it as just something that Mike gets to do. I think we're all going to be able to do this.
I understand that you're also involved in something called personalized medicine, in that some people react differently to different types of medication, and that can be predicted by their genome, so you could tailor medication depending on a person's genetic traits. In fact, I think we have a slide about that. Can we see that slide, please?
Yeah, so if I may, what you have in the middle there is the population, the diverse population of humanity. And when I give a medication, I have a rough idea what might happen to the patient. But let's be honest, I think we've all had an experience where the drug doesn't work exactly as planned. Sometimes it'll work exactly as we hoped. Sometimes there won't be any effect at all. Sometimes there'll actually be a toxic effect instead of any kind of efficacious effect, and sometimes there won't be, there'll be a toxic effect as well as a good effect. And you can see all those different combinations around the side there. I think the important thing is, in your genome are variations in your DNA that allow us, in many cases, to predict whether a certain medication is more likely to be toxic for you or more likely to work well for you. And this is possible because we understand how drugs work, and drugs are metabolized usually by the liver or the kidney. And if you have genes that are in the liver or the kidney that have small variations, you may, for example, absorb a drug faster than somebody else or slower. That difference in absorption will lead to a different experience of the drug. Similarly, the drug, when it reaches its target, that is to say the protein that it interacts with, there may be variations in that target that make the drug bind either more tightly or less tightly, and that could lead to a different experience of the drug. There are drugs that everybody in the world has tried. Codeine is one commonly known drug where there is a huge variation in the population of whether codeine is effective or not. And most people don't realize that variation is because of their genetic background.
What is the ultimate thing you're trying to learn about in genome research? What is the holy grail of all this?
Well, in my mind, the goal would be to know the genome so well that when a child is born, based on their DNA sequence, we could predict all of the diseases that they're at risk for. It wouldn't be deterministic, it would be probabilistic. And what I mean by that is the following, that based on a genome sequence, you'd have a certain chance of getting a certain kind of disease. And we would like to know that, again, the genome so well that if there were a change that came out, you would be able to make that prediction. And that would be my goal.
Is there a possibility of fixing somebody's genome so they're no longer vulnerable to that disease?
There is. There's an entire field called gene therapy or gene intervention, where we try to introduce genes that have been mutated to the point where they're not functioning and they cause a disease. Sometimes you can reintroduce, using a variety of viral or man-made, human-made vectors, introduce a fixed-up version of the DNA into enough cells that the organ starts functioning appropriately. And we're looking at this for some very rare diseases where there's a known genetic variation. And there are trials now in humans that are quite promising. So I think that in our lifetime we may see focused correction of a severe human disease.
Are there any long-term ethical issues with this? So, for example, let's say we have the ability not just to fix a genome so you're less susceptible to a disease, but so that you can get new traits altogether. Can we breed people to specifications, and would that be a good thing?
Well, that's obviously a controversial area of genetics. I think no one will argue with curing horrible diseases through gene therapy or catching disease early and figuring out how to deal with it. But modifying behavioral traits, modifying things that you're talking about enhancements of someone's physique or what have you. I think that's a very controversial area, and one that one would go through that process very slowly, shall we say, to be able to determine what's proper ethically.
And importantly, that's a societal decision. I think some of these technologies may make these things technically feasible, but it's just because it's technically feasible doesn't mean you should do it. And also, just because it's technically feasible doesn't mean that the technologists should be the ones deciding if it's right or wrong. So for those kinds of questions, it's all voices that need to be heard, and there has to be a societal discussion about where we draw the line, if we draw a line, at manipulating genomes.
Okay. Because there's a possibility that everybody might be typed at birth, and somebody might look at your genome at birth and say, well, doesn't look like you're going to amount to much.
I'll go one step further. People will probably get their genome sequenced before birth so that you can predict, because now there's DNA circulating in pregnant women that has the fetal DNA, and in principle it's possible to sequence their DNA before birth. So you'll actually be able to predict the sorts of diseases they might be at risk for or might have at birth, and you'd be able to catch them early.
And if I could even push that further, Mike and I are aware of companies and technologies where you can take an embryo, you can take one cell from the embryo. It turns out that if you do this early enough in development, it doesn't kill the embryo. Just that one cell it can be made up for, and then you can sequence and make measurements on that single cell to learn things about this embryo that is now an 8-cell embryo, just 2 or 3 cell divisions since the moment of conception. It sounds amazing, and it is amazing, but there are companies that are doing this.
Is there any possible downside to this? Is there any way in which this knowledge might turn out to be harmful if it's misused?
Absolutely. I mean, I think a corollary to the technologies that we develop can make amazing saves, if you will, is that if used improperly, you could imagine targeting soldiers from the other side who might have certain genetic features and saying, we're going to build viruses that are specifically designed to attack those people, all kinds of things that you can imagine. And again, I think society needs to decide if and where to draw the line. The problem is sometimes the bad guys, if there are bad guys out there, might want to have a different set of motives and operating principles. So I do think that this is a powerful technology. We're obviously committed to using it for the benefit of humankind. That's our #1 priority. But I think it would be putting our head in the sand to say it's impossible for somebody to use it for nefarious goals. I don't know if you agree.
No, it's true. I guess right now I think we're certainly very focused, as Russ says, on using it to improve human health for the benefit of people. To me, it's a no-brainer. This is what we should be doing. But like all things, you should act responsibly and accordingly. The area will be regulated, no doubt, to try and minimize these sorts of things, but you do have to watch for abuses.
Does it look like there's a pretty open field, like this research is progressing nicely, no major obstacles in the way, got enough funding, and you just see a steady progress until all of the goals are achieved in the not-too-distant future?
Well, there's still many challenges ahead. We are not perfect at interpreting the genome. It's still a huge challenge to get to the goal I mentioned, where if a base change occurs, if there's a change in your DNA at one spot, what will it be its consequences? That's going to take quite some time to be able to sort that out. It's also going to take a lot of resources, even much more than we have now. So there still are funding challenges for many of us. We'd like to be able to enhance what we're trying to do. It's an incredibly exciting area. It's incredibly fast-moving, but it's going to take a lot of effort to be able to get to the point I mentioned. I also think we want to get at this issue Russ raised earlier about not only understanding your DNA, but how it interacts, if you will, with the environment. So that, for example, should you be a pesticide worker if you're at risk for Parkinson's? The answer is no, based on some interesting studies from one of our colleagues at Toll Butte. But we want to understand that better. Which kinds of things should you do and should you not do based on your DNA? And that's going to take a lot more research. It's a very open frontier, a huge challenge ahead.
You know, I'm interested in statistics and computer science, and another big challenge is there are variations in the genome that are only going to be present in 1, 2, 10, 50 people. Very rare. It's going to be impossible to study those statistically. We like to take 1,000 people or 10,000 people with a variation and compare them to 10,000 people who don't have the variation and learn the differences. That has worked well. But when you're talking about a rare familial or even personal variation, those kinds of statistical analyses are not going to be possible, and then we're going to have a much deeper understanding of how to predict these through computation. And so there's a huge opportunity and challenge for computational analysis of rare DNA variations for which we're never going to have enough examples to do it in a straightforward statistical way.
So I think your research is mostly computer modeling, right? You're modeling the behavior rather than doing wet work?
My research, absolutely. We do not have, the wettest thing in my laboratory is the espresso machine. Okay. And everything else is in the computer. We have cluster computers. We have, all kinds of computational software. It's been an amazing time because when you have 3 billion bases or then 1,000 people with 3 billion bases, this is fundamentally an information processing problem. It's why it's exciting also to be here in Silicon Valley where those kinds of numbers don't scare people.
Now, if your genome makes you susceptible to a disease, does that mean that you have a faulty gene, there's something wrong with it, or there's a normal variation, and so different people are normally responsive to different things, so one individual might suffer, but somebody will be resistant, so the species as a whole will be hardy and able to survive?
Well, that's what it's thought, that a lot of these variants, if you will, are common, and so some of them help certain groups survive adverse conditions and others are deleterious. And the classic example is sickle cell anemia, where individuals with a certain genetic trait that makes them susceptible to sickle cell anemia, they're actually more resistant to malaria. And there are many other examples of that as well. So some of this stuff can be protective, but and then some of it's deleterious in the wrong situations.
One of my favorite examples on that note is diabetes. So we know that there's this problem with obesity, and obesity is leading to an increased epidemic of type 2 diabetes. Why is it that so many people get obese and so many people get diabetes? Well, 5,000 years ago, when droughts happened, if you had an ability to retain calories and get the maximum efficiency of the food you ate, then you were the one who survived at a time of drought or famine. That was an incredibly valuable capability. Now, fast forward to the era of McDonald's and plentiful food, that's not the greatest adaptation to have in terms of your ability to cling to calories because there's too many calories around. And so diabetes probably, and there's been work in this area, arose as a very useful way to avoid starvation, and now we're left with the consequences.
So whether your genomics work for you might depend on the environment. Maybe you're well defended against something that's never going to happen in your environment.
Exactly.
Yep.
Exactly. And so you always have to think back. As a geneticist, we do this a lot. What was it like 10,000 years ago as a hunter-gatherer, say in Africa or in America, and what were the big things? Starvation and infection. So much of our genome has been optimized to not starve to death and to avoid dying of an infection. Again, fast forward, we have antibiotics, we have a lot of food. It's a different set of issues now that are killing us. And we have not evolved to respond to these new threats.
We have about 30 seconds left. Any last comments?
Well, I want to say the other area that genomics is having a huge impact that we didn't talk about is cancer and undiagnosed disease. And so I think these are areas that if you have cancer, every cancer is different. You will, in fact, want to get your genome sequenced to be able to get the right drugs for the cancer. And genomics is also having a huge impact in undiagnosed disease as well for trying to solve some of these. But there's huge challenges ahead.
Those are here today. I agree entirely. That's what we can do today.
Okay, great. And we're going to have to leave it at that. I'd like to thank my guests, Michael Snyder and Russ Altman. Thank you for watching. I'm Marty Wasserman. Visit our website, www.futuretalk.net. See you next time.